I am Dr.T.Sreelatha, Assistant Professor of Botany. Today we discuss about Anthoceros. Systematic position of Anthoceros. Division Bryophyta, class Anthoceratapsida, order Anthoceratales, family Anthocerataceae, genus Anthoceros. So the Anthoceros thallus grown into moisture soils, shady soils, and in hill station Visakhapatnam, Mahbubnagar hill stations in AP. The Anthoceros thallus distribution in all over India. So coming to the vegetative morphology, this is the external structure of the thallus. So the thallus divided into dichotomously branches, unequal dichotomously branches. So it is look like one flower shaped structure, and it is in green color, dark green in color, and it has upper surface has small green color spots. The spots are called mucilage cavities. So the mucilage cavity, nosta cavities present in the mucilage cavities. So long elongated structure present. That long elongated structure is called sporophytes. So the thallus having long elongated sporophytes, so this type of thallus is called hornworts. So long elongated sporophytes here, we can see the picture. So this type of thallus is called hornworts. So coming to the internal structure of the thallus. So internal structure of the thallus is divided into the thallus is divided into two parts only, epidermis and middle part, middle region. So epidermis again it is divided into two parts, upper epidermis and lower epidermis. So upper epidermis it is also single layer, and lower epidermis also it is also single layer and made up by parenchyma tissue, and lower epidermis having rhizoids. So here the only smooth rhizoids are present here, rough rhizoids absent, and scales also absent here. Only smooth rhizoids are present. So the smooth rhizoids are absorb the water and minerals, and it gives to thallus and help to fixation into the soil. So middle part is called middle part. So middle part, total middle part made up by parenchyma tissue. So here all parenchyma cells are similar to all parenchyma cells are similar. So all are equal. So the parenchyma cells are totally equal in shape. So each parenchyma cell having one chloroplast, and each chloroplast have one pyrenide. So here presence of pyrenide is algae character. Presence of pyrenide is one of the algae character. So this is not bryophytic character. So here middle of the region total made up by parenchyma tissue, and the parenchyma cells are all are in equal in size. So each parenchyma cell having one chloroplast, and each chloroplast have one pyrenide. So pyrenide is the presence of pyrenide is one of the algae character. So this is not bryophytic character, it is algae character. So they contain chlorophyll cells, so they are participants in the photosynthesis and prepare the food. So in middle of the region some cavities are present, that cavities filled with mucilage. So that cavities are called mucilage cavities. So nostoc colonies are present in the mucilage cavities. So here some cavities are present here in the middle region, and the cavities filled with the mucilage. So this type of cavity is called mucilage cavities, and the mucilage cavities filled with the nostoc colonies. So here the nostoc colony giving to the nitrogen to the Anthoceros. So first picture and the second diagram we can see the cavities. Nostoc colonies are present in the cavities. So nostoc given the nitrogen to the thallus, and thallus given place to the nostoc. So here this is also internal structure of the Anthoceros thallus. So here second picture we can see the second picture, mucilage cavities present here, a nostoc colony present in the mucilage cavities. So coming to the reproduction. So reproduction takes place by two types, vegetative reproduction and sexual reproduction. So coming to the vegetative reproduction, here first one is fragmentation. So fragmentation means the thallus accidentally divided into some pieces, and each piece develops as new Anthoceros thallus. So this is called fragmentation. Second one is tubers. So the thallus produces at the edges of the thallus. So the tubers are separated from the thallus and they produces new Anthoceros thallus. And gemmas also present on the thallus. So here there is no midrib. So the gemma cups are present on the total surface of the Anthoceros. So gemmas, here the gemmas also participates in the vegetative reproduction. So the gemmas produces new Anthoceros thallus. So here the thallus with gemma, we can see the clear picture here, gemma cups. So the gemmas are separated from the thallus and they produces new Anthoceros thallus. Next, coming to the sexual reproduction. So the sexual reproduction, male sex organ is called Antheridium and female sex organ is called Archegonium. So here antheridial. So the primary initial produces antheridial chambers. So here the antheridial chambers having 3 to 4 antheridiums. So 3 to 4 antheridiums. So coming to the development of antheridium. So antheridium, the antheridial chamber produces primary initial. So the primary initial cell divided into two cells, upper cell and lower cell. Lower cell, basal cell is called stalk cell, and upper cell is called antheridial mother cell. So basal cell divided into sometimes, and they produces one small stalk, and the stalk is attached with the antheridial chamber, and antheridial mother cell divided into so many times, and they produces one round shape structure, and the outer side produces jacket cell layer, and that layer protect the inner part of the antheridium. So many cells are divided into the middle of the antheridium. So each cell forms chambers. So each cell forms chambers, and each cell change into androspore. So the androspore has here we can see the clear picture here. This is antheridial chamber having 3 to 4 antheridiums. So each antheridium three to four antheridiums. So the androspores, the chambers, antheridial chamber produces antheridial initial divided into upper cell and lower cell. The lower cell forms as a stalk, and upper cell antheridial mother cell divided into outer layer of the jacket cells, round shape structure, and outer layer, the jacket cells layer. The jacket cells layer protect the inner part of the antheridium, and it forms so many chambers, and each chamber contain only one single cell, and that single cell convert into androspore. So this androspore in a spiral shape structure, and it has two flagella. So this complete formation of antheridiums, antheridial chamber layers ruptures, and each antheridium jacket cell layer ruptures, and the androspores comes outside, and they swims for some time with the help of flagella, and they search for the archegonium. First picture, we can see the first picture. This is androspore in spiral shape structure, and it has two flagella. So next one is Archegonium. Female sex organ is called Archegonium. So the Archegonium, the initial change into Archegonial initial. So this Archegonial initial divided into 4 cells, and 4 cells. Middle of the cell is called axial cell, and three peripheral cells are covered to the axial cell. So here the axial cell was covered by the 3 peripheral cells. So the 3 peripheral cells again divided into more times, and they forms one flask shape structure, and outer they forms jacket layers, and they forms one flask shape structure. So middle of the axial cell, again it is divided into 2 cells, upper cell and lower cell. Upper cell is called neck canal cell, and lower cell is called venter canal cell. So neck canal cell, again the neck canal cell divided into 4 cover cells and 6 neck cells. So 4 cover cells covered to the upper side of the neck part, and 6 neck cells are covered to the neck part only. So here the neck canal cell divided into 4 cover cells and 6 neck cells, and 4 cover cells covered to the upper side of the neck part, and 6 neck cells covered to the neck part only. So then lower cell, venter canal cell, again divided into 2 cells. Upper cell is called venter cell and lower cell is called X cell. So this is Archegonium. Archegonium, complete it is in flask shaped structure, and it is covered to the jocket cells, and upper side 4 cover cells covered to the neck part, and 6 neck cells are covered to the neck part, and below the neck cells venter cell is present, below the venter cell X cell is present. So this complete formation of Archegonium. So the 4 cover cells and neck cells and venter cell, so these total cells are ruptures, and they produces one sweet chemical, and the androspores, they swims for some time with the help of flagella, and they attract with the sweet chemical, and they comes into Archegonium, and they enter into the Archegonium from neck part, and so many male gametes enter into the Archegonium, but only one male gamete fusions with one female gamete, and they forms one zygote. So here the androspores attract with the sweet chemical, and the fertilization based on the sweet chemical. So this fertilization is called chemotaxis. Next, the female gamete and fusions with the male gamete, and they produces zygote, and the zygote divided into so many times, and they forms one long elongated structure on the thallus. That long elongated structure is called sporophyte. So coming to internal structure of sporophyte. This is the internal structure of the sporophyte. So the sporophyte is divided into 3 parts. Foot, seta here seta is absent. So meristematic tissue present here. Third part is capsule. First part is foot, second part is meristematic tissue, and third part is capsule. So here in the seta absent here. So first one is foot. The foot attached with the thallus and absorb the water and minerals, and it gives to the capsule. So it is made up by parenchyma tissue, and 4 to 5 layers are present, and it is attached with the thallus. And second part is seta. Here seta is absent, and meristematic tissue is present here. So the meristematic tissue continuously adds the new cells to the tissues so the sporophyte continuously to grow for a long period. So here the meristematic tissue continuously produces the new cells, continuously adds the new cells to the sporophyte. So the capsule continuously to grow for long period, for long time. So the sporophyte is look like elongated structure because of the meristematic tissue. So third part is capsule. So here the capsule is present here. So we can see the clear picture here. So third part is capsule. This is one elongated structure. So three outer layers, 3 to 4 layers of the epidermis is present both side. 3 to 4 layers of the epidermis made up by parenchyma tissue. So these 3 to 4 layers of the epidermis present both side of the capsule. So the epidermis has chloroplast. So the epidermis have stomata. So stomata contain chlorophyll cells, so they participates in the photosynthesis. So this sporophyte, Anthoceros sporophyte, partially depended on the thallus. They depended on the thallus only water and minerals. So only absorb the water and minerals, and they participates in the photosynthesis because of the epidermis cells contain chlorophyll cells. So here they participates in the sporophyte participates in the photosynthesis also. So here the sporophyte partially depended on the thallus. So here 4 to 5 layers of the epidermis present both side, and the epidermis contain chlorophyll cells. So the chlorophyll cells participates in the photosynthesis. And next, middle of the part is called columella. So this columella present. So the epidermis made up by parenchyma tissue, and middle of the part is called columella. This columella made up by parenchyma tissue, and it is present 16 vertical rows. So 16 vertical rows, and this columella present in the middle of the sporophyte. So the epidermis present outer side of the sporophyte, and the columella present middle of the sporophyte. It is in 16 rows, and it is made up by parenchyma tissue, and the columella present 16 vertical rows, and so the columella, function of the columella, it transfer the water to the capsule, and it gives to mechanical support to the capsule. So the columella made up by parenchyma tissue, it is in 16 rows, and it gives to mechanical support to the sporophyte, and also transfer to the water to the capsule. So middle of the in between the epidermis and columella. We can see the clear picture here. In between the epidermis and columella, spore mother cells are present here. Middle of the part is called columella, and outer layers are called epidermis. Epidermis present both side of the sporophyte, and middle of the part 16 rows part, that is called columella, and columella gives to mechanical support to the capsule. And in between the capsule and epidermis, spore mother cells are present. So the spores mother cells are two types are there. One mother cells are fertile mother cells, another one is sterile mother cells. So two type of mother cells are there, fertile mother cells and sterile mother cells. So here the sterile mother cells change into pseudo-elatus. the sterile mother cells convert into pseudo-elaters. So the pseudo-elaters in narrow shape structure, and they did not have any spiral thicknings, so they are called pseudo-elaters. So the pseudo-elaters helps to dispersal to the spores. So when the time of the dispersal, the elaters are helps to broken the sporophyte, and helps to pushed out to the spores. So then coming to the fertile spores. So here the remaining spore mother cells are fertile mother cells. These fertile mother cells in diploid stage, so they undergoes to meiosis division. So they undergoes to meiosis division, and they divided into 4 cells, 4 applied cells. So this stage is called tetraspore stage. So this stage is called tetraspore stage. So this stage is called tetraspore stage, and the tetraspores are separated from each other, and they forms haploid spores, single haploid spores. So the spore mother cell undergoes to meiosis division, and it is divided into 4 cells, and that stage is called tetraspore stage. So the tetraspore stage, the cells are in applied stage, so the tetraspores are separated from each other, and they forms one single haploid spores. So when complete formation of this single haploid spores, outer epidermis layer and columella layer ruptures, and the spores comes outside, and they enter into the water or moisture soil, and they forms one new Anthoceros thallus. Here columella, meristematic tissue, the spores are enter into the soil or moisture soil, and they forms one new Anthoceros thallus. So here germination of the spore. So this is the Anthoceros life cycle. Thank you, children. Thank you.